Hi, I'm Vladimir Sandler, co-founder and CEO of Chimeric Pharmaceuticals and Bait Biotechnology. This is company overview. So Bait Biotechnology, Chimeric Pharmaceuticals developed a novel platform for the development of radically new treatments of existing and emerging viral infections in certain types of cancer. This platform is being spun out as a subsidiary of Bait Biotechnology. We filed foundational patent application protecting this new technology. We're located in West Harlem in Manhattan, New York City. So Bait Biotechnology is a company that is being spun out that enjoys access to R&D and manufacturing infrastructure of Chimeric Pharmaceuticals, and also access to network of advisors, board of directors, and business and fundraising experience, as well as talent of its parent company. So this is our current team that includes many distinguished individuals, including two Oscar Prize winners, Mark Feldman for anti-TNF antibodies for rheumatoid arthritis, and Mark Shepard for herceptin. He got his prize in 2019, and we have also our clinical advisors and others. So what are we developing? We're developing antivirals and brain cancer treatments. All our treatments are based on so-called bait-based programming or redirection of macrophage using novel synthetic proteins. Synthetic advantages of our product candidates are we can develop, and we're developing single therapeutics targeting multiple viral infections. Our product candidates have long shelf life and can be easily deployed or administered at ambient temperature, and our brain tumor treatments can easily cross blood-brain barrier to target brain cancers. So for viral infections, unmet needs are clear: global pandemics and biological warfare. We are offering Chimeric Bait Receptor and its derivatives, Bait Macrophage Engagers, as a means to prevent and combat infections by pretty much any known or emerging virus. Our goal is always to prevent future pandemics and render virus-based biothreats ineffective and provide American warfighters with an overwhelming advantage in case the adversaries decide to use bioweapon. So the market is very large. Our current global market for antivirals is about 61 billion, projected to grow by 33 to 89 billion US. Unmet need in brain cancers is also obvious. So we're targeting brain tumors that include gliomas. Our solution is Chimeric Bait Receptor that will be used in combination with programmed hematopoietic stem cells to cross blood-brain barrier to eliminate brain tumors. Obvious goal is to develop reliable treatment of malignant brain tumors, including glioblastomas. So why do we need new solutions? We have vaccines, antivirals, and antibodies. However, all of them are sensitive to emerging viral infections. The main advantage of CBR-DME that we're developing is that it's insensitive to emerging viral infections. There are also other advantages. So what is CBR? CBR program immune cells to target viruses or cancers. This is being used as part of cell therapy. And what is BME? BME is cell-independent constructs, basically biologics that can be used as off-the-shelf drugs for the treatment of viral infections or certain types of cancer. So how does it work? What is bait? Bait is a chunk of protein that the virus needs to bind to enter the cell. So CBR is a synthetic receptor that consists of the bait. Consists of the bait jump into this protein that virus binds to, and the macrophage receptor. What is BME? BME is a bait, linker, and naturally occurring ligand for a macrophage receptor. So we achieved proof of concept for SARS-CoV-2, COVID-19 virus. We demonstrated that synthetic virus being that we can attack and kill synthetic virus using our CBR. We're finishing BSL-3 studies demonstrating that CBRs against SARS-CoV-2 work against real infectious SARS-CoV-2, and we have a bunch of product candidates that cover a wide range of viruses, including dengue, Zika, Ebola, Marburg, and chikungunya, and all these viruses are targeted by a single construct. We have, I think, a very smart way to administer our drug candidates. So we use mRNA, self-replicating mRNA that provides us longer lasting expression, high density of CBR, and high production of BME, and we can administer this using this approach where we have polymers that are mixed at the field with mRNA and then inhaled by the patient targeting cells in the upper respiratory tract. We also achieved proof of concept for CBR administration for brain tumors. We engineered hematopoietic stem cells, injected them into immunocompromised mice, and demonstrated that they generate a number of blood images as everyone else. And we also demonstrated that they generate human brain microglia that resides in the CNS of these mice. So, in summary, we have a novel proprietary technology. We achieved proof of concept. It's directly applicable to U.S. Department of Defense and HHS services objectives. And technology is ready for translational science to develop and test products. Bait Biotechnology has the facility and people to execute the program. Thank you so much for your attention. No, if you, for example, think about SARS-CoV-2, SARS-CoV-2 binds to ACE2. As long as SARS-CoV-2 binds to ACE2, no matter how the virus mutates, this works. Also, different families of the viruses use the same protein, bind to the same protein to enter the cell. So we can take that protein, identify the portion of that protein, usually extracellular portion that is needed for the binding, and then use it as good.
Thank you. And the second one is the question of you explaining about the
Right. So the biggest challenge in treating brain cancers is EBV, right? So we demonstrated that if you take human hematopoietic stem cells, inject them, transplant them into immune-compromised mice, you end up with all kinds of blood lineages as you should, B, T, and so on cells. However, in addition to that, when you slice the brain, you see that there is a progeny of these hematopoietic stem cells that are microglia. So now we are engineering hematopoietic stem cells to have CBRs that would target surface markers of glioblastoma.
And do you see efficacy in models?
We are not yet in vivo. We showed it only in vitro, and it's kind of very early. We're trying to expand this approach to cancers.
I have a follow-up question about GBM. I'm curious, why you go to the trouble of, hematopoietic stem cell transplant when, ostensibly it seems like you could, if you know the surface marker on GBM, and they're already microglia in situ with the tumor, and you said that the construct here will generate across the blood-brain barrier. Is that correct, or they don't?
No, the constructs themselves do not go to the brain.
So they have to be
cells,
programmed
yes.
into.
I mean, there are ways to cross BBB without this approach, but this is too cumbersome. This is a natural way. You take HSCs from the patient, engineer them, you have an unlimited source of them, you give it IV, and it goes.
And as sort of a first proof of concept, if you consider those sort of ways to circumvent the BBB to sort of like convection enhanced delivery or other things that are sort of more cumbersome but possibilities for GBM treatment to sort of get proof of concept for the CDR specific to the target.
So we demonstrated that we can inject mRNA encapsulated in polymers through the nose, right? So when you do it, actually some of it goes to the brain. And so we will look at this, but we're not yet there. And it's not universal. We don't know how well it goes and where it goes.
So sorry, we only have about 10 seconds left,
Just a shout out for Vatislav's company. I'm with Prevail Partners. We're a committed investor. And anybody who'd like to join us as an investor, please see